I just got back from Costa Rica too, a couple weeks over Christmas, and I'm really interested in ecosystems and plants and fungi as well. I did do landscape design for 20 years while I was doing mycology. So when I went to see Rick, I said I would not work anywhere else in the upstate if it isn't for an ecological based landscape company, because I worked for Henry Ford, Serena Williams. I worked for all the rich and famous in West Palm Beach, and it was spray, 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 kill, kill, kill. When I came back, I went to Clemson and went back to school for microbiology. It was the greatest thing I ever did. Greatest thing I ever did. So started Mushroom Mountain, which is a company in 1996, and then I sold it 2 years ago when I split with my ex. So that was one of the best things I've ever done, because now it freed me up to do some things where some research and some exploration. I really felt like I wasn't being creative anymore, that I kind of hit the wall, and it was just a production type of job. You'll see what's going on now, and this is the reason why I can't sleep at night. So I hope you toss and turn in your beds after you hear this and see this. The mushroom kingdom is absolutely amazing. It's the largest, second largest kingdom on the planet next to insects. So there's 5 million species of fungi out there estimated, and we've only found and ID'd like 160,000. So this is a huge kingdom, and a lot of them are invisible. They're yeast, they're things hiding, and we're going to look at all this. So I got a lot of slides, so I'm going to go fast. This is the tree of life as we kind of know it. What we think is right today is disproven tomorrow, but as far as we know, this is little branches of life. Fungi are here, animals are here. We share a lot of common things with fungi. Mushrooms create heat. They produce carbon dioxide, so they require oxygen, and they also sweat, and except they swim through their own sweat. We don't do that. Well, maybe in August. But this is not accurate because there's interkingdom interactions. We're not a single organism here. I'm not trad. I'm a coral reef. I'm like a composite of mostly bacteria and a lot of different things. So if you remove everything else that's not trad will die, right? Same thing with plants. So when I'm out in the woods, this is me morel hunting. Does anyone ever go morel hunting? Yeah. What's your favorite spot? Okay. All right. So what's your favorite spot? So morel season's coming up within a few weeks. It cool off a minute, but I love going out and just tuning into nature, turning off the phone and disconnecting, because I learn out there. I don't learn a lot from Googling, what I need to know or what I can discover. You got to get out there and look for it. This is a mushroom that was found in Savannah, Georgia, several years ago. This is Macroscope titans. This was found by a school teacher who mailed it to me by request so I could clone it, because it was very rare in South Carolina. This one is usually found in Costa Rica, Mexico, Florida, but it's starting to creep its way inward, as our climate gets warmer. This one weighs 50 pounds, and you could sit on it. It will hold your weight. It is edible. Every mushroom is edible for 30 minutes. It's 2 feet across. In Costa Rica, they get 4 feet tall and 3 feet wide. They get absolutely huge. This mushroom is involved with the collapse of ant colonies, tropical ant colonies. So we're talking about multiple levels of biology here. It's not just, hey, we see a mushroom. What is it doing? Why is it there? What is it feeding on? What is its mechanism in the environment? Mushrooms are like first responders. When you want to know what a mushroom is, you can take a spore print. This is one of the methods you use one of the steps that you would use to identify a mushroom. Put it down on foil or paper, and it produces a color, right? That's how they reproduce. Those spores are microscopic, and each one of those spores has half the DNA that it needs to make another mushroom. So that's why they produce millions, and they land next to each other, and then they have to germinate And then they have to find another mate of the same species out there with those 5 million plus species producing billions of spores. What are the odds, right? That they land, germinate, and mate. And then they produce mycelium, what's like a little island, and they vegetate and take over their environment. So you've seen mushrooms on the ground, the forest floor. You've seen them on logs. You've seen them on trees. That's what they went through. They went through germination. They mated. They met on the dance floor. They liked each other. All right. And then they took over that little part of the world. And the mushroom fruits, when they run out of, when they think that they're running out of food, so they always want to be feeding and colonizing. But then they start to feel threatened. A mushroom is a defense mechanism. That's all it is. Right. So whenever you see a mushroom, that means it's scared out of its life. It feels like, hey, I better produce more spores and get more out there to produce more islands and more babies somewhere else, right? Because this place is done, right? I can feed on it, but the battery is gonna run dry, right? Mushrooms produce heat, carbon dioxide. They do sweat. You can see the heat, the hot red tips here. That's called hyphae. A group of that is called mycelium. And in that chemical sweat that they swim through, I actually think I'm a mushroom, by the way. So you swim through your sweat, all right? And then in that sweat are enzymes, almost like digestive enzymes. They combust molecules, organic molecules, and they bust them apart, and they use those atoms as food, as a food source. And that's how we were talking about breaking down diesel and atrazine and herbicide, is because when they run out of food, fungi get desperate, and those same enzymes work against molecular contaminants, man-made contaminants. It's unbelievable, right? So they can feed on many different things. Mushrooms can be predatory, submissive. They were just really good at orchestrating, right, and balancing the environment. They ultimately want to heal the soil. Now, I am a little bit biased. There's also pathogenic fungi. There are honey mushrooms. There's parasites that attack trees. There's parasites that attack us. There's fungal infections for animals. But they're really good at regulating that, right? It really depends on the situation. So when I find a mushroom that I like, or if I want to propagate it, I germinate them in the lab. They'll mate, and this is mated mycelium. It's perfectly symmetrical and it's pure. But it's not what happens in nature, because there's mixed colonies out there. This is a zoo animal. This is like a zebra at the Greenville Zoo, or there's no competitors. So really, you're just feeding it, and it's losing interest in survival at that point. But I can take that fungus and I can expose it to the elements, right? Just like in nature, because those mushrooms that are growing on the logs, it's not sterile in that wood, not at all. There's other fungi, there's other molds, there's bacteria, there's protozoans, you know. So this is what happens when it gets defensive, because mushrooms are opportunistic, right? And they're territorial, and they like to conquer land masses, and they like to mine the resources until they're gone. Then they find a new place to live and start all over again. Does that sound familiar? Right? So it's really not hard to think like a mushroom, you know? I think I'm in the final stage. So this was an experiment. This was just an accident in the lab. When I was a fellow at the EPA, I left some plates out, and I got this green mold on a plate, and then that beautiful symmetrical arc of mycelium that was a zoo animal responded, and look what happened. So then it surged, it stopped growth all the way around on the other edges, and it surged like an army over the top of that green mold, like that. It's defending its territory. And what else do you see? It's the production of sweat, which are novel compounds dialed up to destroy and kill that particular fungus on demand. That's wild, right? So they're capable of producing their own biological weapons against other forms of biology. And so I pit off these bacteria. I'm not sure if I'll show this later or not, but I do these gladiator matches. It's probably why I'm single. I do this Friday nights. What are you doing? Uh, Shiitake mushroom, oyster mushroom, and this is what I'm watching, instead of watching the Super Bowl, I'm watching my little match. And and then you see these droplets and you get to see like the interaction. So this is what you don't see under the bark, right? Or under the grass. You don't see them bumping heads because really mushrooms do not like each other. I hate to say that. It's not Care Bears and Skittles, right? It is just the nature, and even the same species like us are competitive. They're different strains, they're different ecotypes. So an oyster mushroom over here and here, if they were to meet, they would do the same thing. They would try to kill each other, right? They're very competitive for their space. Mushrooms also produce chemoattractants, which are interesting. So these are exudates that a particular mushroom, all mushrooms can produce them, but they're absolutely beautiful. Some of them are octanols, or what I'm looking into now, which are very interesting, is chemoattractants for bugs, specifically to use them as chemical lures for bugs that are problematic in agriculture. So we can take something like I'm working on one now for stink bugs because I love tomatoes and I hate those damn things because they get on my tomatoes and they pit them all up. But I'm working on a yeast, which is a fungus. So this gets out of my comfort range a little bit. That it's not a mushroom, but it is a fungus that produces an octanol. And if you want to catch fruit flies, the strongest octanol producing yeast comes off of a mango. So if you want to make a fruit fly attractant, use mango and a little bit of soap, soapy water. They'll go right in there. They cannot resist mango yeast. Also, in the presence of nematodes, without the presence of nematodes, it's very linear, very cottony mycelium. But in the presence of nematodes, fungi also produce these little circular snares that you see here. They're loops and they are traps. And they're trigger traps. So if a nematode is nearby, these dumb nematodes stick their heads through these things, and the mycelium in real time will contract and strangle the nematode, kill it, and then sweat out enzyme, a very corrosive enzyme, and then they'll eat it. So they will actually eat the worm for dinner as a protein source. I do children's shows too. So it's always a little kid. Does that happen to people? I'm like, yeah. No. But there's a micrograph. It's just so cool looking, right? These are little inflatable rings, like like the kids wear in the pool. Oof, just like this. Just man. And mushrooms are soil producers. That's what they do. And nothing irritates me. Well, a lot of things irritate me, but one of the things that really irritates me in the fall is I was driving to a tree conference in Kentucky, and I was driving through like Louisville, and I see in the urban environment everyone's lawn leaf bags out on the curb, like in the little Home Depot Lowe's bags, and they're full of leaves to be taken away. And I just really got a little pissed off. I'm just like, there's a reason they're called leaves. You leave them there. That's what they're for, is the trees drop them, they decompose, the fungi are there to convert that into mycelium, and then the worms eat that, and on and on. So that is the food web. So we're pretty destructive as far as the food web and the soil web. And what's attracted to that mycelium is worms, right? I went to the International Worm Symposium about 15 years ago. It was interesting group of people, right? It was like 500 crazy worm people, right? And I was the only mushroom person there. It was like Comic-Con for worm people. And I was just interested in composting mushroom stuff with worms. And they said, one of the guys, oh, it doesn't work. Well, I went back and I bought a pound of worms from someone in Easley or Pickens. And then now it's a chapter in my book called Mycovermicomposting. It's amazing. So having mycelium in your garden around your plants is a good idea because it attracts other organisms, it unlocks soil nutrients, and then you have this fertilizer that's happening, right? Look at this beautiful soil that I made. within a couple weeks. Love my worms. Love that worm compost. Mycorrhizal fungi. These are fungi that are bonding with symbiotic with trees and plants, and this is a symbiotic relationship. And there's a lot of mycorrhizal species here in North America, maybe 4,000 to 5,000 ectomycorrhizae, which are mushrooms that you can see, and those are, I can name a few. Chanterelles. Anyone? Chanterelles, porcini, boletes, black trumpets, morels, partly, truffles, on and on. You know, they keep going. Very important for our ecosystem. You know, chipmunks, a chimpmunk's winter diet is 60 to 80% underground fungi. 60 to 80%. You take the trees, you take the fungus, you take the chipmunk away, right? As much as 60% of the plant's photosynthesis comes down and as exudates into the rhizosphere, which is the root system of the tree, right at the tips, the new growing tips, and that feeds the fungi and the bacteria in the soil. And then in return, the mushrooms are their interface, so they are bringing in nitrogen and phosphate and other minerals and giving it to the tree. So they are symbiotic that way. So they're partners, right? And they're paired for life. And also they offer disease resistance to the plant. So the plant on the left, if there was high phosphate, if you're using a natural or excuse me, man-made fertilizer with a lot of phosphate in it, then this relationship will not exist because the fungi are very sensitive to this, and so is the plant. If the phosphate levels are like in the, I think it's like 5 to 8 parts per billion, it will not allow the fungus to enter the root system of the plant, right? It would prefer just to get the fast food through the drive-through from you, you know? So this is the root system you want. You have disease resistant on the right. You have nutrient transport, and you have the fungus delivering nitrogen and phosphate at the rate that that plant needs it and at the time that it needs it. How do we know exactly when to drop that on there, right? So the disease resistance and the mycorrhizal associations have been going on for millions of years. It has not changed much, so it's a perfected system. And a lot of fungi are going underground all of a sudden. They're finding out that a lot of the guild mushrooms that we're looking at now are starting to evolve to go underground. They're hiding like they know something's coming. Right? Carbon trading, water transport and retention, root pathogen resistance. Show that, you know, with the sweating, the metabolites. It's in their best interest to protect that plant for their future. They want that tree to grow bigger. They want it to drop leaves, nuts, branches, wood falls. Here's a whole nother food source for the future of that fungus. All right. We know what that is. Indian pipe, a plant without what? Chlorophyll. So where does it get its energy? It actually taps into a fungal mycelial system. So there's a mycorrhizal mushroom. It's red, and it is interacting with trees, and they're symbiotic. But this little plant taps into the system and gets free cable. Right? We don't have cable anymore. What am I talking about? So there's all kinds of mycorrhizae. Like I said, ectomycorrhizae, there's over 5,000 species. A lot of these, unfortunately, there's only a very small number of mycorrhizal fungi that are used in the ornamental and the forestry industry. And I think that's a mistake. And Rick was talking about these nurseries and things like that. If we had a more diverse regionally biospecific like inoculum of trees and plants that had more truffles and more chanterelles, because the ones here that I'm showing you, Pisolithus and Tinctorius and all those that the forestry department uses, they produce poisonous little round mushrooms that'll bust up concrete. They don't really do anything. They speed up the rate of the tree, but where's the biodiversity, right? Yeah.
Just when when I started working with native plants, I knew about mycorrhizae. I knew the benefits of it. And if you're going to do native plant landscaping, you better be using mycorrhizae fungi and not chemical-based fertilizer. So in 30 years, I never used chemical fertilizer. Use this stuff. And back in the day, for trad, and before you go now buy these products, I used to go buy root flag from grading companies, and they thought I was crazy. What? Nobody wants this stuff. I said, "Yeah, I know who wants that stuff. I want it."
Yeah.
And it just was a cabinet.
I'm writing a new book, and one of the chapters is how to create your own, or how to propagate your own mycorrhizae for your plants. Listen, go out into the woods and just dig up a hole, dig up some soil around a tree that's healthy, that has mycorrhizal fungi around it, around the plant that you want. You know, if it's little blue stem or whatever, do doing like a transplant of those root fragments, and there's even the cut root fragments, the new plant will reach over and grab it, right? It's not that complicated.
Pretty broadly by the breezes and wind.
Of ectomycorrhizae, yeah, but endos not so much. They don't produce a lot of spores. Yeah, it's a lot. Some of them don't. So yeah, there's the killer. And so I'm a damn good gardener and a plant propagator, and so I know that I keep this out of my garden because I know it's a fungal killer. It kills mycorrhizae. But listen, once you get mycorrhizae into your soil and your garden, it exists there. So after about 1 or 2 years of inoculating your garden, it's on root fragments and it's natural. But when you start putting this, and if you're putting fungicides in the soil, I'll talk about that later. Yeah, that's on my list too. So be careful, or try to eliminate that. That's not the way nature had it all set up. Nitrogen fertilizer increases community, like fungal diseases, and it's just a domino effect. Once you take the fungi out of the ecosystem, it becomes bacterial dominant, and you need a fungal dominant soil all the time. This is conventional soil. If you were to slice these roots and put it under a microscope, it would be dead. It would be, there would be no mycorrhizae in it. Whoever is cultivating this field of corn or cotton or what have you, these plants are addicted now to whatever. They're now in the cycle. You can break the cycle. It takes a minute, but you have to put fertilizer on these plants because there's really, if you really need them to be productive. And if there's no root resistance or any, if you're spraying with antifungal compounds, then it becomes open. Then there's no endophytes, which are fungi that are inside the leaves that I'll show you that protect the plant. So you're essentially killing its whole immune system, right? You're putting your plants, they look good, but they're on life support. So then you start seeing outbreaks of diseases. You start seeing Phytophthora, and you start seeing diseases that are coming in from other regions on plants that are shipped in, from out of state. They come here, and are if these plants are under cultivation and they are fertilized and they are not mycorrhized, then they may not stand a chance. So I remember working with Rick that when we would go out and do these plants, we would use special species of fungi and bacteria to combat Phytophthora and also mycorrhizae, and trichoderma, which is a green mold, which is a mycoparasite. It's like everything. You're putting the biology back, right? You're giving the plants what they need in that soil. Microremediation is a form of bioremediation using fungi. This is fascinating. It's nothing new, but it's underexplored. There's not many people working on it. Fungi are really good at mitigating biological threats. They can stun and kill E. coli. I've made mycological water filters for Jamaica and Haiti, out of sand and fungi. And E. coli are 50% protein by weight. It's just a snack in the eye of a fungus. It's just food, it kills it and it's protein. It'll inactivate it and kill it and eat it. Chemical pollutants, I don't need to list any more than herbicides, oil spills, diesel, coal ash, things like that. Mushrooms will hyperaccumulate heavy metals. That's where it gets tricky. When someone says, "Hey, can you clean up this coal ash spill in North Carolina?" I'm like, I don't know. Hold on a second. That could be a challenge because the mushrooms could hyperaccumulate the heavy metals, and then what? Right now, you have it, concentrated in a little edible packet that wildlife or people can eat, and then it gets back into the food web. So it's not for every solution. Years ago, I collected some oyster mushrooms when I was out at dinner. I literally tripped across the stump and found them in the middle of February. It was snowing, and I cloned it, and it actually became one of the best strains that I use for remediation. And so I put herbicide on the plates, in the plates, nothing else. And I did the labeled rate, and it completely colonized the plates. And then, so they're starving to death, so they're going to eat anything. And then I did double the labeled rate, and it did it again. And then I did triple the labeled rate just to see what it was like trying to feed my black lab. Like, when is it going to stop? You know, bottomless pit, but it did. It slowed down, and this is at triple the labeled rate. And look at that, at day 10, it just absolutely hit the wall. It just stopped. And then I was like, okay, so I'm going to backtrack, find maybe where its limits are, where its thresholds are. But then I only came back 2 days later, and look at this. So this is what I like to do on Saturday. It's like, wow, whoa, what is it doing? So what we figured out is these fungi have huge key sets, genetical key sets that organize, and they can create cocktails of enzymes in those, and they swim through that. So it was learning from that. Now on those tips, do you remember those little hot red tips I showed you? That's called a Spitzenkorper cell. You'll never remember that word again, right? But it's a little tiny little a bundle of like microtubules, and it can sense what's in the environment, and then it reprograms the mycelium to produce something else. It's almost like a chromatography, like a little beam of light that can shine on that and say, you know what? I think we need to turn these genes off, turn these on, and create this, or we're in trouble. All right. And that's what it uses to kill those molds too, and those bacteria. It's all makes sense. It's all on that front edge of mycelium. And interesting enough is they don't even have to touch it. They can get a whiff of it from an inch away, and still, it still like exudes something into the agar, that gel, and diffuse it and kill bacteria. It's wild. So I do plates like this, and I put them on part per million, part per billion, different colors, just so I can see it. I did this for my book, so it was prettier. Well, you need to see that, that also I started just dumping that oyster mushroom strain on just about everything. I'm like, well, it's like Mikey, like what else can it eat? Right? If you knew who Mikey was, then I know what age bracket you're in. Who doesn't know where Mikey is? Exactly. The commercial from Life cereal. Yeah. Terrible cereal. So yeah, so I started dropping it on like motor oil, and I started started to just feed on that. There was nothing in that plate, by the way. That's gel. That's seaweed gel, non-nutritive, 10 weight 30 right out of the bottle. Right. They're finding fungus. This is another one. This is not new. This is years old. Pestilotopsis, microspora. This is not the only one that they found. They found more, but they found that this is a fungus that lives in. Well, they've also found it living in Algerian ivy in Israel, but they found it in a plant in Brazil, inside of a leaf, not even producing a mushroom, where it was actually breaking down pure polyurethane in the lab. Pure polyurethane, right? The hopes is that we are headed towards finding solutions in nature that could possibly reverse some of this plastic problem that we have, right? Make things more biodegradable. Packaging that is self-destructive would be absolutely gorgeous, wouldn't it? Right? Take that bottle water or the bag, put it in the compost pile, and poof, you know, mushrooms come flying out. That would be my house. Mushrooms are strong. This little bundles of mycelium is here on the right. You see those like those little threads. Those are millions of layers of of hyphae in there, just little bundles and twists. And what's interesting is that they hold the soil together, which is important. I don't think anyone has seen or needs a reason to know that holding soil where it needs to stay was any more important than October, right? When it went away, right? And I've been up to Asheville, I've been up to Barnardsville, and I've seen things just gone, just blown away. These threads, and this is actually a King Stropharia mushroom. You can grow it. It's a good edible. But if you have this in your garden or on a bank or a creek bank, it holds 20 to 30,000 times its own weight, right? In areas where the hurricane floods came ripping through, they said the soil was gone in some areas, but when they saw wood chips that were fully colonized that were holding the banks, right? So that's the stream repair that we need to do. You got to have those fungi there. You got to put those wood chips back on the slopes. You can't pile up the trees. You can't clear a piece of property in Greenville County or anywhere and pile up the trees and burn them, right? That should be against the law. We should chip them up and put the carbon back so the mushrooms can do their job of soil creation and holding the soil where it needs to be held, right? And no more important than soil and erosion control here. I'm no stranger to the different methods and products that they use on slopes. Does anybody know what they typically use? Mesh, yeah, and it's plastic, right? Anything that'll just hold it for a second and grow grass. That's what I was told by the researchers. They said that all they want, these construction people, just roll it, grow grass. Who cares if it has plastic in it, right? Or even this straw, which they can lay down straw and then they spray like a tar substance and then it sticks it together like glue. Great for the environment. I see a different method. I see hydroseeding this with fungi and some different organic material, agricultural byproduct. And then the mushroom can hold it like glue. There's one fungus I have that it holds about 1 million times its own weight. And my mom found it. That's her legacy. She found it in Key West, Florida, in the lapping surf of Key West Harbor. So its salt tolerance was really high too. And that's a fungus that can break down oil and it has salt tolerance. Do you see what? That's huge. And that happened right after, she found that right after that big BP oil explosion. Where's that BP? I shouldn't say names, but probably. Deep water or something. Do you have a question?
So for erosion, how long does that take from the time you put soil back on it?
I mean, if you put organic compounds out there and you spray mycelium, especially the one that I have, this tiger sawgill, I mean, it'll be plasticized within days. Plasticized, like it'll just like that. It's a phenomenal fungus. We're actually using it at Clemson Engineering Department for composite materials. I've made bricks out of them. And when they try to cut this with their bandsaw, it broke. Yeah, I laughed. Yeah, like, I think that's pretty damn cool. Go mushrooms. Try to cut my babies in half. Endophytes. This is an exciting area that I haven't had enough time to get into, but I will this year. I have to because I have to write the book about it. But these are fungi that live in the plants. These are the invisible threads. These are that 4 million that we don't know about, right? Every plant, if you go outside with a little hole puncher to any plant and you punch it and you dip it in alcohol and water over and over and you think you've sterilized it and you put it on a Petri plate, it's in there, right? And it comes out, right? So, but the coolest thing is that the reason your plants are so brilliantly green and healthy, that they have this endophyte that they coexist with in their leaves, stems, and roots. So they have mycorrhizae in the roots, and they have these endophytes and all this other tissue that are trying to protect it from other fungal pathogens. You know what they are: powdery mildew, downy mildew, leaf spots. It just on and on, right? So what's so romantic and poetic is that the plant, when it produces seed, and that's why seed saving is so important of healthy native plants, is that those plants, as they grow, it deposits the endophyte into the seed for jettison. So the next generation has what? Not just the plant genetics, but also the fungal genetics of the endophyte that it needs, that really benefited that plant. So we're planting composite organisms. Yeah.
Do those tend to be host specific, or they minerals?
They could be host specific. You know, that there's very little's known about them. So that's why I was so excited to, you know, when I met Rick years ago, like we just started, man. We just started getting crazy because endophytes, I think native mycorrhizae is huge, but native endophytes would be amazing. And I think it's essential in the plant health and their disease resistance. So I do have a lab in Greenville. I'd be happy to collaborate. But that's cool. And so yes, your composite organisms, they are in between the cells of the plant. And that's why when you start to see fungal diseases like this, I'm interested in, actually, you see this fungal infection here and then here. I would take a sample here of the good tissue and take this one and put them on the same plate and see. That would be like my Wednesday night gladiator match. See what's going on. But you have to see, you know, you could take, that's the front line. So that makes sense, is if you're going to find something that's really putting up with this, try to isolate this and see if it's active. And then you can generate enough inoculum to put in with the new seeds. So as soon as the seeds germinate, you can put them in your soil mixture, and then the endophyte could enter the that root radical that comes out. Or they've also found that you could spray the endophyte, if you have a solution, up under the guard cells and into the stomata, and then they'll go up and thread up into the leaf and get into the plant. Whew. Good luck sleeping, right? I told you. It's nuts. There are companies that are working on putting different endophytes in plants, right, in Europe. So I'm looking at all these diseases, and a couple years ago, Clemson gave me some botrytis from these strawberries, and there's so many diseases out there that when growers see this, what are their options, right? What are your options if you're a strawberry grower and you start seeing these outbreaks? You spray. You know, sclerotinia root rot, it goes on and on. These are just mismanaged fields. They're not organic. Look at that bare soil. If there was a little bit of mulch or wood chips around there, it might stand a better chance. And a friend of mine, a mentor, Walker Miller, who is from Happy Berry Farm. They were familiar with Walker. He was one of my mentors. He got me into mushrooms. So he really did, when I was 20. And he had a problem with Botchоспhaeria on his grapes. This is just a picture of some muscadine stuff. But he said, I need to find a solution for this. So I came out and I punched his leaves on his grapes, and I found a green mold, a green mold that was living in it as an endophyte in his leaves. And we propagated that. And then I grew it on matchsticks, and we drilled into his grapevines when they were dormant. And then that triggered a phytochemical response. And the next year he had no Botchоспhaeria whatsoever. And that was not my knowledge. It was a study that was going on in New Zealand that was brand new. And he goes, I want to try it. I'm like, damn, how about that? Huh? That was simple. Isolating, you know, antagonists from the farms where there is a problem, because mushrooms, fungi got to find a solution. The Earth will lose 40% of all insect species in the next blank years. Double jeopardy. Probably now, yeah, 22. The largest kingdom. Think about it. Is that a problem? It's a huge problem. It's a big problem. Why is that happening? A lot of you would suspect that the number one culprit would be insecticides, and it's not. But this is the native plant group. So I'm going to say, what's the number one cause of insect decline? Loss of habitat. Right? Loss of habitat, number one. Pesticides, human, number two. And then so I started looking at different insecticides. First was herbicides. I showed you that with atrazine. Then I started looking at different natural fungi called entomopathogens. These are fungi that attack living insects, and they're out there, right? This is Bovaria. It's a white powdery little fungus. It's got a very broad appetite for different insects. And so I find these are aphids. This is a winged aster, I think. And Todd Elliott, I don't know if anybody knows Todd and Doug Elliott up in Asheville, but his son found this and sent it to me. And these are fully mummified aphids. And so this group, this little colony of aphids got into some nasty stuff, right? And so this is a good strain. So I could take one of these aphids in the lab and I can make millions of this stuff. If I wanted to try to propagate this fungus and train it to be more target specific on aphids, I could. That was my lecture 2 weeks ago in Georgia, in Atlanta. It was mycodefense, which was how to create novel novel entomopathogens. And these are, it's a way of, now we don't want to, we don't need to propagate anything that's going to attack pollinators or winged insects like this, but just as an example, we found this one in the Francis Marion Forest up above, is it Ondo? Is that Francis Marion? Yeah, I used to live in Ondo. I walked into the woods and I found 20 of these wasps upside down in the leaf duff under a little tree. And all of them had a mushroom growing out of their neck, like right there. Everybody touch that little soft spot right there. Feel it. Imagine a mushroom coming out. 20 of them that were buried. And so these are called cordyceps mushrooms. And this one I found at Mepkin Abbey, the monastery, the Abbey Monk's Corner, when I did some work for them. And I was photographing another mushroom and I see this little club sticking out of a rotting log. I took my knife and popped it out. And here's a cicada nymph that is fully mummified with a mushroom growing out of its brain. Oh, man. Right? What a find. And this was the first time it was ever photographed. I had no idea. I sent it to an editor, Fungi Magazine. He goes, holy crap, Trad. You know, I was like, where'd you find it? You know, I was like, I was just hunting mushrooms, looking for something else. And it was right there on the log. This is one of my favorites. Everyone in this room, everyone on this call, if you're in the upstate, you can find this one. It's so easy to find. It's all over the upstate. If you look under American holly trees and beech trees, you can see them, especially this time of year where there's no leaves, like, or the leaves are, you know, not as green. This is an ant that has been parasitized by a fungus called Cordyceps unilateralis. And this fungus infects the ant when it's alive. It threads up into the body, into the nerves, and then it surrounds the brain of the ant. And then it secretes chemical information and it steers the ant around the woods and up the tree, and it bites, it makes the ant bite into a tree. You see the bud scar? Nice little entry point, huh? So its mandibles pierce that, kills the ant. The fungus thread comes out of the mouth of the ant and enters the living tissue of the plant to harvest more water and nutrients. And then the mushroom pops out the back of its head, out of the brain, straight down towards the pheromone laden highway that the rest of his buddies are using, right on top of him, right? That study was done by Harvard, Dr. David Hughes. And so they're out there, right? There are fungi out there that we can use to be at least try to maybe be more target specific for specific insects, not using these broad spectrum pesticides. That's not going to work. Not going to work anymore, right? Look at that one. If you need a Halloween costume, look no further, right? You know, biting down right on the main arteries of that leaf. They found mandible scars in leaf fossils that were millions of years old, 120 million years ago. There were fungi attacking the brains of ants. Yeah. This particular species of a cordyceps, it's very narrow target specificity. So I'm hoping to get to one that I found. This one is one growing on scale insect that I found on a sweet gum. Just walking around looking up at tree limbs, you know, and just see them. So you can find these if you're out looking at plants. Turn the leaves over. I keynoted at the Cullowhee Native Plant Symposium last year, and I always take a group out, and every single person found a cordyceps because I showed them where to look. They just didn't know it. Everybody was freaking out, right? And that's what I hope for, is if you go and look this year and turn over leaves and look for bugs that have something suspicious that don't look normal, like fuzz or white or little horns or something coming out of their body. I mean, if you saw a person like that, you'd turn them in, right? And so this was my first cordyceps that I ever found, courtesy of Rick, because Rick said, "Oh, you got to go where the rare plants are, and I'm not going to tell you where the spot is." So I put off going there for months and months and months, and I found this 5 minutes into my walk. It was my first cordyceps, growing out of a log. It's about the length of your eyelash, and it popped. I didn't know what it was. I saw a little hole, so I took my knife and I opened up the log, the rotting log, and look at this beautiful thing. And so I took it to Clemson first, entomology. They're like, this is Camponotus pennsylvanicus, or if that's pronounced right, but it's a black carpenter ant. And then I sent it to Harvard to Dr. Hughes, the expert who figured out the whole zombie ant thing. He goes, "Where'd you find it? We haven't seen that species of fungus on that ant since 1907 in any documentation or any herbarium." And he goes, to top it off, this is a queen. This is a queen to be. And so I've found maybe 7 of these in my lifetime, and it's been a minute since I found them, right? But they've all been on queens. She was huge. She was like the size of a hornet. So what I did was I took my dental tool that I got at the Pickens flea market. I think that you're selling dental tools. Oh man. Anyway, took my Pickens dental tool and I cloned it. I opened up her abdomen, took a little piece, and then I was able to clone it. And then I reinfected new black carpenter ants. And then I knew I had the pathogen. So then I started messing around with it. So this was probably like a Monday night. These aren't the fire ant farms I made. This was a stock photo because I had to figure out, I was like, I want to watch what's going down. You know, this is what I do during the week. So I can't just put it in soil. I want to watch the behavior. So we made fire ant farms in my house, and I reintroduced that fungus to the fire ants. And within 4 to 7 days, that's what the fire ants looked like. So that's the way a fire ant should look like, by the way. Bad hair day. Right. And that is beautiful, isn't it? That's just beautiful. The way, see how it's coming out of the hinges, the legs and all that? That's what you're looking for. A lot of fungi attack insects post-mortem. So it looks like it could have just been a dead bug that just got attacked by something. But when you see it coming out of the cuticle, the armor of that insect, it's coming out from the inside, right? So then I started looking at pesticides, and then I started looking at the bottom 2, which were Solenopsis and Camponotus were very closely related. So I was thinking, well, maybe that stood a chance. And I saw this chart before I introduced that to the fire ants. So I thought it was a good Hail Mary to try anyway. I tried it outdoors. I had 8 acres. I treated one pile with this new fungus that I found, and within 24 hours, those ants were gone, and we didn't see any fire ants for 5 years with one dose. And I've done other studies too. Clemson's been helped me. They gave me almost all their stuff. I'm not sure if they're doing fire ant research anymore, by the way. They gave me everything. It's kind of weird. The honeybees, those little varroa mites that are on the backs, luckily that they've, there's a university in Texas that found a metarhizium, which is a fungus that attacks the varroa mite that is sitting on top of a bee. It doesn't hurt the bee at all. Not at all. That varroa mite is responsible for meal feeding and injecting viruses into our bees, right? There's alternatives other than pesticides that are going in our hives, all right? There's so much pesticide pressure on bees. So, I've been out there collecting as much as I can. There are some commercial products that are on the market. MET52 was owned by Monsanto, and then, it was owned by Novozymes, and then Monsanto bought it, and then they discontinued it for 7 years, and now it's back on the market again. So I don't know why it took a 7-year break. But anyway, those are products that are out there that are alternatives. And this product works on varroa mites and brown spotted ticks and termites. Are termites essential? We should kill all the termites, right? Yeah, they're very essential. They're very essential. I know you don't want them on your house and your structures, but hey, why not treat just around the wood? Why not spray this on the wood? Why not grow it into the wood? See? What do you think we're doing at Clemson? We're doing some weird stuff over there. And this was found up at Yellow Branch Trail up near Issaquah Falls. I mean, once I found one, I think they were everywhere. It's like then the mushrooms were finding me, right? And then I'm like, okay, well, I'm going to put out maybe most wanted posters for mushrooms that eat oil or that eat, that are salt tolerant or that are on a bug. And that's what I was really doing out in Atlanta 2 weeks ago at that conference. Was like, we are going to solve this problem together because I need everyone in here to also look for bugs. Name a bug that you want to find, right? Fully mummified, and I can give you a handful. So wherever I travel around the United States or abroad, I'm like, tell me what the problem is because I don't know. All right. If I'm in Costa Rica, is it attacking the cacao plant or the banana or the avocado? What is it? Look for that bug that's mummified because it's probably here mummified. And then we can amplify it and use it. Like the emerald ash borer, right? Wouldn't it be nice to find this little booger, right? There should be a lottery for this, right? There should be a price on their little heads. These were found at the Cullowhee Native Plant Society conference. Literally, I lectured on Saturday, and this girl took all the pictures of the cordyceps that the group found the day before. So they were all in the crowd, and they got to see their cordyceps on the main stage, right? So we were finding all these mummified bugs and caterpillars. It was awesome to see a group of beginning, mushroom collectors out there. Really, I mean, I couldn't get them out of the woods after the ID. They were like still wandering around the picnic area, right? They're like, what time is this over? I'm like, it was over an hour ago, but I've got nothing to do. Aren't they gorgeous? So if you see little structures like this, don't just pull them, maybe dig up a little and you might find a little prize underneath. I'm trying to speed up as fast as I can. Am I okay? Okay. Okay. Okay, good. And then there's the problem of this is the spotted lanternfly is on my list. And I had some newer slides, but these are fine. These are just fine, but these are devastating, and they are headed this way. I knew a lot of growers and up in Jersey and friends in Jersey. They're already there. They're attacking the grapevines, and when they eat something, they're just going to eat that. These fields look like they were just napalmed. You know, they were just absolutely black and dead. 3-4 generation vineyards and grape growers just done, just fried. These are the only way to combat these right now. Right now, they say is to spray like heavily. And I honestly, I tried one of the sprays on a couple on someone's grapevine, and I hit it, and they're so waxy, and they roll, they just jump, and they pop, they jump, they're gone. They're very hard to kill. And lately they've been using like glue traps around the trees, and that catches woodpeckers and chipmunks, and it's awful. Don't look it up. I did it for you. But then someone found these in Burke County, Pennsylvania, because they went to my lecture, just like you're watching today. And they said, hey, I think I found something suspicious. It looks like spotted lanternflies that are fully mummified. Do you see what's coming out of their legs? Hello. That is beautiful, right? That's what you need. That's what we want. We got to look for those stink bugs. We got to look for those, Japanese beetles. We got to look for those bugs that are fully mummified. Because this is what I was teaching 2 weeks ago, is that if you can find one, you can collect more, and then you can take this one and amplify it because it's inoculum. It's like a dirty bomb in the room for these bugs, and infect 10 more, and now you have 10. And then what are you doing? You're like me, you're blending up the bugs in a blender on a, Thursday night, sifting body parts. Thursday. The earth has lost half the all wild animals in the last 40 years. So we cannot solve a problem with the same thinking we used when we created them. The world is it's a mess. I'm an optimist. I say I'm not a pessimist, but I'm pretty optimistic this ain't gonna work. I've lived through so many hurricanes, man. Like when I lived in Florida and South Carolina, and they get stronger, and this is evidence up here that, dude, when does a hurricane hit Asheville, right? It's crazy. I saw it coming too. I was like, these trees aren't ready. I stocked up. I knew it was going to be bad when I saw the trajectory. We got to be more prepared for this. We got to prepare our trees. We got to make our forests stronger. We got to make our soil more biologically diverse. We got to keep the wood chips down. We have to understand that fungi are like first responders. They're here to help us. So we have to form an alliance with the fungi and the plants at the same time, right? It takes over 500 years, 600 years to make 1 inch of soil in a healthy forest. Is this a healthy forest in the front? Upstate Greenville, Trees of Greenville did this study. We were at the same meeting years ago when Dr. Shai was giving the speech. There's areas up here that were the soil depth, the average soil depth was 14 to 15 feet. Topsoil. Now it's 4 to 5 inches. And I did the math. If we did nothing to put that back at that rate of 500 years to be low, it would take 75,000 years to put back that soil if we did absolutely nothing. And we lost it in 100. So here's our friends, right? Here's what we need. We need reforestation efforts, these clear cuts, these stumps. This is easy to do. There's fungal inoculum, or just let it show up. Put the chips down, build it, and they will come. That's a mushroom movie. Every time they say that, I'm like, that's it. Put the welcome mat down, right? Put it down. That's why we got to keep the carbon here. We can't just throw it in a landfill, put it out on the curb, right? So this is a forest that I see in my lifetime, and my daughter and my daughter's, and all of our families. We want them to walk through fields like this, right? We got to return it back. Because as far as I know, there's at least one of these out here. Right? Until we find the next one. But let's fix this one. Anyway, that's it for tonight. I can keep going on and on. So you want me to repeat the question? In here? Okay. That's my squeaking water bottle for use at home. Right here first, and we'll go back. No, go ahead.
Is mainstream.
To what extent?
Yeah.
Not that much, not that much. Mainstream, not conventional mostly. Organic farmers are catching on. Mycorrhizae is been around a while, but I think the issue is that the inoculants aren't really, some of them are not effective. They're products that are sold in liquid, and they may be very poorly regulated or even checked. I think it's good to, if you can, to get a product analyzed so you know that it has viable mycelium and things in it. All right, but who can, not everybody can do that. Get it under a microscope and look for viable mycelium or spores, right? The mycopesticides is the two products. MET52 is one. The other one I can mention is called Mycotrol, MYCOTROL. It's an okay product. I'd rather you use that than a pesticide, molecular pesticide, but it does have a broad range of target specificity, broader than I like. So I think there needs to be more. Now, what's interesting is that they're finding out that, you know what an endophyte is now, right? It's inside the plant. They're finding out that these entomopathogens, which are the pesticide, the fungi that kill insects, specific insects, can actually exist as an endophyte. So imagine if we could take the fungus, the more target specific fungus, put it in the plant of choice. Like if I found something for stink bugs, I would put that in my tomato mixture seeds or get them in there, and it might be able to give some resistance to that. That's interesting. And I think there's patents filed about that already by some of the big ones. I'll come back to you in a bill in a minute. Yeah. I've written one so far. Yeah, it's over there. And then, yeah, we had it. Organic mushroom farm. I'm trying. It's been really busy, but it's been exploratory, which is good. Sometimes I feel like I'm not working on it, but I am. I'm always working on fungi. So, yeah.
The possibility of mycoherbicides. I'm thinking fake buttercup. We need a fungus that eat the hell out of fake.
Yeah, absolutely. And it also scares the out of me too, because where does it stop? Fungi are self-replicating organisms. And when they run out of food, they might go after something else.
Yeah.
So that's like someone asked me, well, can you find a fungus that kills Bermuda? I'm like, yeah. And yeah, but what would that do to the Bermuda, grass horse industry? Exactly. I'd have to move to Costa Rica willingly. Please take me with you, Rick. But yeah, mycoherbicides are good, and I started. It raised an eyebrow when I started to find fungal pathogens on like poison ivy and things like that, or English ivy. Wouldn't that be cool? Right? They're there. That's just like nobody's working on it. I wish there was more. It's like there's one me and a couple of mycologists that don't work on it, that kind of stuff. The other things I've been working on are novel antibiotics. It wasn't even a part of my conversation. I think that would be fascinating, but it doesn't fit our talk tonight. But I could take a disease sample and inject it into a bag of sterile mycelium, and it creates a novel antifungal or antibacterial compound that you can dilute 10 to the 6, which is 1,000,000 times. So one sample of that botrytis on that strawberry could produce 520 gallons of a more target-specific fungicide in 24 to 48 hours. That, to me, is the future of agriculture. That sounds fun. Maybe that's what Einstein was talking about. It's just so nature can do it, you know? If you look deep into nature, you'll find the solution for everything. That's what he said. Oh yeah. Oh yeah. Yeah. I painted you the rosy picture. And the picture was, yeah, do fungus also attack animals? Of course they attack us, you know. Of course they do. Athlete's foot. Do you know what causes dandruff? You want me to scare the hell out of everybody right now?
Nathan Attenborough has a YouTube video on this type of infection.
Yeah, I mean, there's the look at what the white-nosed bats, and I mean, I did a paper on that in school, and you know, it may not have been the fungus that it could have been a virus, and then their immune system dropped temperature, then the fungus sets in secondary. You know, they still don't know. But yeah, there's fungi that definitely attack. Yeah, I did some medical mycology classes, and they're pretty gross. Not for this group, not before dinner. Another question, yeah.
That's something that is available for sale.
No, not yet. I have to go out and get some more collections, and I know that's why I said I won't tell anybody where they are, because I've only found them in 2 spots in the upstate. No, I patent nothing. I filed 2 patents for the medical device and the process, and that's it. But I think there's a lot that there's an EPA loophole. Thank you for paying for my education, EPA. The EPA loophole that states that if you find a biological within the state, you cannot sell it, but you can use it in that state. So if we find it in here, we can propagate and we can use it. You just cannot sell it, and you cannot you'd have to register it as a pesticide. You have to get it registered as a pesticide. It's naturally occurring as a real, well, for now, an EPA loophole. So that's why I'm encouraging, that's why I like to lecture and travel. It's going to say, listen, you find it in your state, and then create a little bioregional lab for $1,000, and everybody in the whole state can use it and benefit from that, and maybe even share it. Like in Pennsylvania, those now 100 spotted lanternflies. Can you imagine what we could do with that? We could just multiply it like sourdough bread and just give out friendship bread to everybody. I'm serious. That is how easy it is. It can be. Because I've had that fungus since 2017. And Cornell has it too. And I don't see anything going on. I'm not picking on Cornell. I'm not, there's a lot of states that are doing that, but the chemicals are still there. And I don't think I mean, what are you going to inject chemicals into a tree, right? For $200 a tree every year? And then are you going to kill other organisms? Does that pesticide go into the flowers? Does it go into the nectar for the bees? I mean, it's like, what's going on here? It's just not working. So, but yeah, I think of myco-herbicides all the time. Going back to your, I was thinking about kudzus and things like that. That was my first, a kudzu or.
You found an organism that's created a fungus that created a herbicide.
Right.
Would you have to separate that? You have to isolate that compound from that fungus, or would you buy fungus and
I would rather isolate the compound, because I wouldn't want the self-perpetuating organism in the environment to go after something else. Yeah, that's a good idea. I'm glad I thought of it. That's in the far back.
Yeah, that's small scale for like paralyzing gardens. Is it more effective to try to introduce some sort of fungus or to just set up a building blocks like wood chips, compost, that sort of thing, and let whatever comes from it?
I mean, that's a question of, I think at least let whatever comes come, right? But if you want some really cool mushrooms in your yard that you can eat, you know, that are really good, like King Stropharia and these others, I mean, that company's Mushroom Mountain. They have my whole library. I sold it 270 species. And there's soil building fungi, there's bluets for the compost, you know, you can and then once you inoculate that pile, you can take some of that, almost like an ember, right? And move it to a new pile and propagate more and more and more. Yeah. Oh yeah, it's doing really well. Yeah, that's why I sold it. To keep it alive.
They have a really nice website.
Oh yeah, I know. Yeah, question.
My question is, I want to make sure that I understand what I think is the really basic part of your presentation.
Mhm.
And that is the genome of mushroom, all of your eukaryotic cells, but these are fungi. The amount of genetic information on any genome is just enormous, but only a small amount is used for a particular organism.
Mhm.
But these organisms seem to have the ability to read the environment and then use genetic information that's been hidden for 1,000,000 years now, and to use it in a way to solve a problem almost immediately, which
Yeah.
very few organisms can do that.
It's very,
Is that basically what
yeah, they're chemically very quick.
so it's a quick epigenetic expression that almost no other organism.
Well, I wouldn't say that, but I would say that there are, back to the Petri plate, and I'm going to say that the genetics of fungi are, that's why I like to isolate wild fungi. The more you keep a fungus in the lab, the more it becomes the zoo animal. And then that genetic diversity deteriorates.
But it's still, it's in genetic code.
Well,
This-
it mutates and it becomes ineffective. Especially if it's replicating itself over and over, and that's called senescing. And those of us in the particular age group will remember Xerox machines, where you make a copy of a page and a copy of that and a copy of that, and by 10 you can't read it. And so when cells start to make mistakes over and over and over again, it becomes ineffective to even feed itself.
So you're
So-
not saying it's the genetic information to consume motor oil
Mhm.
is not hidden somewhere in genetic in the DNA.
Yeah, right.
It is covered over.
It could be. Yeah.
When it's being replicated, it's not red. But all of a sudden, there's motor oil and no other food to eat. And all of a sudden, the genetic information opens up, and now you have new enzymes that are able to be developed
Right.
that can now consume more motor oil or pesticide or something.
But here's another thing is that not it's very ecotype specific because genetically not every, like say every strain of oyster mushroom can do what that one did with an herbicide. It was a very unique individual, right? Just like we all are in this room, we're all humans. Those are all oyster mushrooms. And if I all put them all on the plates, that some of them wouldn't do anything. If you asked me to build a house, I couldn't do it. If you asked me to make spaghetti, yeah, I could.
I'll give you another example.
Yeah.
Birds come from dinosaurs. Birds don't have teeth. But the genetic information for birds to grow teeth is hidden in their genetics. It's just not expressed.
Mm.
You don't think that the genetic information that these mushrooms are using is hidden deep within the genes of the organism? It's just not expressed. And when you create an environment where that organism can now attach its, or attack its new environment, now it's expressed.
It could be, yeah. We're seeing that they're running out of food. They get desperate and they eventually mushrooms will grow teeth.